Okay, good morning, everyone. I hope you can hear me okay. And welcome to this webinar organized by the FHERITALE project. So my name is Antonio Rosato, and I'm Associate Professor of Chemistry at the University of Florence, and I'm coordinator of the FHERITALE project. Hence, my position here to welcome you all, and again, thank you for your interest in this webinar. I'll just give 2 minutes of introduction to the project to provide you with the context. So FHERITALE is a initiative where it's a coordination action, essentially, involving some European research infrastructures in the field of food health and environment. And more specifically, there are 4 participating infrastructures with different centers for each one of them. And the 4 are analyse, IRIN, Instruct-ERIC, which is the infrastructure I, let's say, belong to, and the 4th one is Metrofood. So as I mentioned, we try to cover different aspects from the molecular level to food and environmental aspects, taking also into account health. And briefly, the major aim of the project is to, since it's a coordination action, is to try and get an overview of the services that these or other infrastructures in Europe offer to researchers in order to address the challenges posed by artificial materials that are let's say, released in the environment. And of course, among these artificial materials, the first ones that come to mind are micro and nanoplastics, which is the theme today. And the overall goal of FHERITALE is to see which services are available, which research priorities are, let's say, covered by these services, and then identify gaps. So if there are any important research areas in the field that current services provided by European infrastructures do not properly address, in order to provide European researchers with state-of-the-art option to carry out these investigations. And finally, to provide ideas, guidelines for future developments. So that's essentially the context, and the 3 speakers of today are actually from external bodies from with respect to the partnership, and this is part of our effort to try and involve scientists and experts that can inform the project and the general community about what's going on in the field, which aspects are relevant, and sorry, I was distracted by the chat, and yeah, essentially to give us some more open perspective on what's going on in the field. So just some logistical notes. We have the 3 speakers running, giving their presentations one after the other. We will take after each presentation only one burning question, if there's such a question, because the idea is actually to have more space for a general discussion at the end, addressing, let's say, together the topics of all three presentations. We asked the attendees to please write their questions in the chat, and then I or the speaker will read them, so we don't have to mess around with giving permissions to speak, revoking them, and so on. And yeah, I guess that's that. So, if there's no further points to be made by the other organizers, I suggest that we get started with the presentation. So for each speaker, I have a short bio that I will summarize. First speaker is Silvia Orlandini from the International Committee for Animal Recording. Silvia is a biologist with experience in milk analysis and working on various topics, mainly for the dairy sector. And she's Italian. She's served as the coordinator of the Italian Breeder Association Standard Lab for more than 2 decades. And she's been involved in various committees, including ISO-IDF, the International Dairy Federation, to revise ISO standards in the field. And she's currently chairing the International Dairy Federation Action Team on Statistics. And you see her title already on the screen, so I will not read it. And please, Silvia, go ahead.
Thank you, Antonio, for the kind introduction. And welcome to all the attendees. And I'm the technical manager of ICAR, and as I'm going to present this NGO organization, I'm not expert of the microplastic, but I hope to provide you some input regarding the standardization process that can be very useful for the researcher, and especially to, in some case, adopted by the legislation and the legislator. So a very brief introduction of the International Committee for Animal Recording. We deal with the animal production. It means that we would like to provide universal language, a standardized language for sustainable animal production. And our mission is to provide the guidelines, standards, and the certification to all the tools that we use for animal identification, recording, and evaluation. It means that the farmer or breeders that would like to enter in the program of the hairy herd improvement situation, they can subscribe or their animals or their herd, and they have to use only certified tools. So just to give you a very simple example, each cow is checked each month. The milk is sampled, it's checked the milk quality, the milk amount produced, and all these data are collected during the lactation period, and the cow is classified for his performance. So this is what means to record in animals, and this is valid for sheep and goat or beef producer and so on. So really, because it's involved with the genetic selection, it's very important for ICAR that the results that we acquire are very well standardized and equivalent to each other, because otherwise the genetic selection consideration will be not equivalent and robust. So our organization we have 142 members. The dark blue show you the map. So we cover all the 5 continents. So really we need to be equivalent to each other. But what is most important is that in our ICAR community, we try to condense the expertise and to promote the exchange between the expertise for the livestock topics. So it means that when the ISO has not the standards for some topics that cover the livestock sector, ICAR will produce the guideline and own guideline in own committees. The process is exactly the same of ISO. I mean, in our committee, we try to find the consensus, and at the end, the guideline is published. It was frequent in the past that we use a guideline for some years in ICAR, and after ISO endorsed, and the same ICAR guideline has been endorsed as ISO standard. This because we have a very strong collaboration with ISO committees, and some of our colleagues that are active in the ICAR subcommittee and working groups are active also in the ISO national committee and international committees. As an NGO, ICAR cannot be an ISO member, but we are an international organization under category A. Means a category A that we have access to the documents, we receive the documents, we are part of the committees, and we participate to the meeting. Simply, we cannot have the vote rights. But in any case, we can deliver comments, and we can bring our expertise through our expert. And we are participating in different technical committees. I will not go in details, but so these are the three committees that technical committees of ISO that we are listed as category A, and we are going to be accepted also in the milk and milk products. This is the area where is my main expertise. So, okay. So this standard is a slide that I summarized some benefit in some details of why an ISO standard is important. As many of you, ISO, International Standard Organization, is a worldwide organization, and nowadays cover 173 national standard organizations. In bold, you can see why it's very important standard and how the standards can support different aspects of our daily life. For sure, what is very important that I would like to underline is that each standard that is published is the result of the consensus reached in the committees. It requires quite an effort to write a standard, because I would like to remind that ISO, ICAR, International Dairy Federation, all such type of organization, the work is voluntary. So really, of course, the delegates are nominated by the stakeholders, by the industry or manufacturers or public institute, research institute. So of course there is the interest, but it means that the delegated person have to run the routine work plus the work and the task assigned in this committee. So it's quite demanding, but the great advantage is that if you are a part of such committee, you are directly in contact with the best expert of very specific topics. So you can meet your colleague that provided a previous standard or that provide a specific publication, and can give you can receive directly for the author a lot of information. So this is the best benefit that I receive in participating in such a committee over the years. But what is very important for me is that if you have an ISO standard, the vocabulary, the terminology, the definition, the graphical symbol are standardized. For me, this also is very important, because probably many of you sometimes read a paper, a scientific paper, and you arrive to the conclusion, and the conclusion are done according some criteria or some statistical calculation, and etc. And if you compare this publication with another one that's speaking about the same method, you cannot compare the conclusion because the statistics is different, or the graphical, the symbols are different. So sometimes it's difficult to understand if what is reported in publication 1 or 2 is the same. So the ISO standard would like to standardize also this aspect, and this facilitates a lot because you have not to recreate a new definition or new symbols each time. Sometimes we have problem, especially between European and American word about that. And what is important is that the standard can be used also for legal use. This is very frequent, especially at the European level. What I really like is this picture. This from the ISO website. And the developing standards is like an orchestra, where the ISO is the director of this orchestra, and each musician is the expert and delegate of our national committees. And for sure, if we develop a standard that meet the market request, we facilitate a lot the benchmarking or the market and so on. Many of you are familiar with the CE brand that we have, so it means that it fulfill the requirement described by the CE brand. So this is what ISO is doing, and for sure, what is very important and really important is that there is a very severe aware control voting system. So the ISO member can vote positively, negatively, and can abstain. If the draft is not accepted by the community, the majority doesn't accept the draft, and the comments came back to the project leader, and he has to reply to the comments, and the draft circulate again. So the consensus is really the key word that is important to approve an ISO standard. So it's a very optimistic scenario that in the best case, a standard can be ready in 3 years, considering all the different steps. There are different types of standards. If you go or navigate on the ISO website, you can find the technical reports that can describe, for example, all the methods that are used for a certain analyte and different metrics, etc. It's a more informative, but it's very important because you can start from that scenario and to focus on a specific method. There are technical specifications. Is listed the technical specification, the standard, they are not completely, we have not obtained all the results. For example, we have not explored the entire range, or there was not the possibility for different technical reasons to conduct the validation study or interlaboratory study. I will detail you later what does it means. And the ISO standard, the full ISO standard, is the full method described in all its details, and where the criteria are also specified. And the criteria could be the precision, and I will detail in the next slides. This is an example that could be useful for you. So navigating for your topics on the ISO website, I found these, you see, is a technical report, and that give the state of the art and knowledge on the methodologies used for the microplastic. I use this slide from your platform because especially for this topic on the microplastic is absolutely an interdisciplinary topic. So, and for that reason, it's very important to be very well connected, but to speak also the same language in terms of, as I said, vocabulary, language, symbols, statistics, and so on. Otherwise, it's very difficult to compare each other, the results that we obtain. I summarize here advantages and benefit. Again, the standard has a very broad geographical representative, and we have a multi-stakeholder environment because the ISO procedure describe exactly that when a new work item is provided or a new method is proposed, there is a questionnaire between the stakeholder involved or interested, if they are in favor or how they would like to contribute, etc. And also what is important, that the ISO standard can be adopted also at the national level, and it's really the result of a huge network. In the context of the regulation and the standard support and different levels, societal and environmental, offer the consumer protection, of course, because if there is a standardized method that determines some analyte that could be dangerous for the health, the consumer are more trustly on it because that product has been checked in a proper way. Of course, this is very useful at the market level, reflect the state of the art, and what it's important, disseminate new technologies. And this is very important for the researcher. Sometimes in the university, in the researcher center, the ISO procedure or the standard procedure are not very well known, or because the ISO, sometimes the normal life cycle of an ISO standard is 5 years, and in 5 years all other new technology can be published. But what is important is that in any moment that a new technology is provided and there is enough interest, at least 5 countries are interested in this new technology, an ISO standard can be published. And in any moment of the life cycle of the ISO standard, the standard can be revised. So it's not necessary to wait the 5 years end. And again, as I said, can be applied at the national level and bring a lot of confidence. Regarding just to give you an example, because ISO is international, but we are used to live in the European context, and for sure some market, or as we can describe the European context in this way, where the system and the standard for quality accreditation and metrology are very well established, and okay, there is the demand over harmonization, and frequently we can use the ISO standard in dispute, litigation, etc. But there are some other countries when they are depending from the imported products. They are lacking consumer infrastructure, and this is the system of metrology, quality assurance, accreditation, etc. are not very well developed, and they have not implemented system for regulation. So when we are dealing with such a situation, for sure, this country, the ISO standard can be mandatory, and this country can really benefit a lot from this publication because they have a starting point at least. So, before I spoken about different type of ISO standard, and I said that the ISO standard is a full method, very well described, and with the criteria described. How to obtain the criteria? Criteria could be the precision, like repeatability and reproducibility, could be linearity, could be low detection limit, low determination limit, etc. But to obtain these criteria is necessary a validation study. A validation study means that the same sample are distributed between expert labs, and they receive this sample and they analyze according a specific protocol that has been accepted by the ISO community. The repeatability, I really like to define these criteria. The repeatability means the absolute difference that you can obtain in a repeatability condition on the same material. It means that if you have a sample and the analyst repeat 2 times the test with the same method and the same lab in a short time, they will have, he will obtain 2 results. The maximum difference between these 2 results should be not higher than the repeatability established in the ISO standard. And this is extremely important because the repeatability is the minimum variance that we can have when we do a test. Considering the reproducibility, that is the same, but in the reproducibility condition, it means the same test, the same sample material analyzed by different analysts in different conditions with different instrument, that is the maximum variability, can tell us what is the maximum variability of that method. And this information can be useful to fix the uncertainty. Why the uncertainty associated to each result is so important? I'm sure that many of you knows this picture. If you have a legal limit, this is the line of a legal limit, and if you obtain these results plus minus this uncertainty, that sample is compliant with the legal limit because it's below considering also the uncertainty. The case B, it's really problematic because in this case you obtain results in your lab, but you cannot say that in 95% of the probability these results will be below the, will be below the legal limit. Case C is the same because you are above the legal limit, even if in few cases could be that you are below, but you cannot accept such results. And for sure, case D, you are out. So all this to say how much is important the process of validation, because validation is necessary to define the uncertainty, and the uncertainty has to be associated to a results and to say if these results is compliant with the legal limit or not. Especially when we deal with the health legal limit, this is extremely important. But the standard method, it's not enough. We need the standard methods and we need the reference material. As you know, we have certified reference material and secondary reference material that if you are lucky that Metrofood has exactly this deal, because in the Metrofood system, we are building the metrological traceability. And if we have a certified reference material, the secondary reference material have to be traceable to it, like the clock, like the meter. But in the case of the meter, it's much simpler than in the food analysis for sure. ISO invested a lot of energies in standardize the reference material. Here I listed some of them. And is extremely important. So scrolling on ISO standards, I found this that could be useful for you or not. By the way, could be worldwide to see how this ISO standard has been prepared. And if you can take inspiration from it, and if you can apply such standard in the environment, health, or food environment, food sector. And also could stimulate your attention. This is a technical specification, you see. And if you see that is interesting for your project and for your for FHERITALE, for example, could be interesting if some of you can participate in this technical committee. The technical committee is this one, ISO Technical Committee 6.1. So if I would like to set the scene for the future and what I've seen from your project, for sure you have already prepared an inventory of the method used for your topic, inventory of analytes, inventory of material and substrate. And for sure, FHERITALE is creating an interdisciplinary, very well-connected research environment. And between you, it's necessary, and when you find necessary, if you would like to start and to consider to start a nice or standard trip, you should contact this technical committee. Is located on DIM in Germany, is the National German ISO Standard Organization, and you can at least to bring your expertise and to deal with the colleagues that already discuss this topic. The next slide is really is an example of what I describe in this presentation and in a practical example. ICAR, since 2016, is organizing a proficiency test on milk analysis. One of the most problematic parameters is to count the cell in milk. The cell in milk means infection of the mammalian gland, and has a big impact in the production, in the health of the animals, and so on. And when we started this proficiency test, with this number of labs in many countries that cover all the 5 continents, we have such a situation. You see that the mean was plus minus 20%, with really some labs that have a very, very distant results from the assigned values. This is just a proficiency test. And thanks to a very nice project that we organize in collaboration between ICAR and the IDF, and a collaboration with the European Joint Research Center, we were able to build up together, finding the consensus, and EU certified reference material. We introduce this certified reference material on the market without any obligation. The labs starting to use this certified reference material. They started to adjust the calibration using directly it or using secondary reference material aligned with it. And in only 6 months, this was the picture. Now this is our equivalence, so plus minus 10%. This was a very successful story, and this is an example how the ISO standard plus reference material can bring equivalence around the globe. Thank you very much for your attention, and I'm here to reply to your question, if any.
Thank you very much, Silvia. It was a very nice and interesting presentation. And as I mentioned at the beginning, if you have one really burning question, write it down in the chat. Less urgent questions we will deal with together with the other speakers at the end. So if there's nothing urgent, we move on to the second speaker of today, who is Alba Hernandez. So Alba is aggregate professor at the Autonomous University of Barcelona, and she directs the Genetics and Microbiology Lab, also together with other, let's say, duties that in the interest of time I'll skip. She works on experimental mutagenesis, toxicology, carcinogenesis, and risk assessment, with an extensive record of publications, and currently she works on the long-term effect of contaminants in chronic regimes of exposure, including nanomaterials and micro and nanoplastics, with a special view on generating information on risk assessment that is useful for risk assessment. And she is involved, she is coordinating the Plasticheal project and co-leading the European cluster project CUSP or CASP, I'm not sure about the pronunciation, that addresses the health impacts of micro and nanoplastics, and you already see the picture and the plastics on the screen. So please, Alba.
Okay, thank you. Good morning, everyone. Thanks, Antonio, for the kind introduction and for the invitation. Today, I'm going to talk about the connection between micro nanoplastics and human health and some of the investigation that we've conducted in the frame of the Plasticheal project. It is quite clear that we produce tremendous quantities of plastics per year worldwide. And during the whole life cycle of these plastics that we produce, there are some factors that can fragment these plastics into tinier pieces called microplastics and nanoplastics. There is some controversy on the definition of microplastic and nanoplastics. The one that we use in the project and in my lab is considering nanoplastics, those that are below 1 micron. And the reason why we put this threshold, let's say, is because all these plastics that are below this micron, all the nanoplastics, are the ones that are able, after reaching the body through ingestion and through inhalation, are the ones able to translocate these primary body barriers, reach the blood, and then bioaccumulate in the secondary organs. In fact, we have demonstrated with several studies and also other authors that these nanoplastics, these plastics below 1 micron, are able to easily internalize inside cells from body barriers. You have here pictures from an in vitro model of the respiratory barrier, and here pictures of an in vitro model of the gastrointestinal barrier. You see the nuclei of the cells in blue, and the nanoplastics in green color. And as you can see, those plastics are a lot and are inside the cells, and they are able to really translocate this barrier and reach the other compartment. If we imagine that this reaches the blood, you have here also some examples on how these nanoplastics can also be very easily internalized in the walls of the blood vessels. These are endothelial cells, so the cells from vessels. And again, you see in green how these cells are plenty of nanoplastics inside. And then from the walls of the vessels, they can go inside the cells, the blood cells, and again, a lot of green in different blood cell types. So it is now accepted, and there are many papers demonstrating that nanoplastics can actually reach many organs in humans, and that what matters and what is important and the type of exposure we are facing here, it's long-term exposure and potential bioaccumulation in secondary organs. So this is on the side of the exposure, and then what happens with the effects of this potential exposure. We know, because of the similarities between the nanoparticles and nanoplastics, that we are facing an exposure to materials that have a high surface area to volume ratio. This means that they have a lot of surface to react with the things that are nearby. So if we assume that we are exposed to tiny plastic that really easily go inside the cell and have a lot of surface to interact with lipids, with membranes, even with DNA inside the cells, we are facing here a potential toxicological potential effects. And this is why everybody is concerned, or many different stakeholders are concerned about what is happening or how we are affected by the exposure to these particles. And besides of everybody being concerned, all the effort of the scientific community and a lot of European restrictions and regulation and initiatives that have appeared regarding or trying to protect the population from these plastics, the situation is still that we do not know enough to really perform a complete human risk assessment. So we are still exposed from many sources through ingestion and inhalation of these plastics. And we have no clear idea that we are really safe because of this exposure. There are many reasons why we don't know enough, but the key questions that we need to solve are the ones that you see here. We still don't know very clearly or very specifically how many microplastics, micro nanoplastics do we have in our bodies, and what are these types of plastics that we have. And we don't clearly know if these specific numbers are a risk to our health. And the reason why these questions are still not answered are in part because we lack validated analytical procedures to really be able to measure micro nanoplastics in external matrices, in the air, in the water, and in the food that is contaminated, and also in human samples. And this affects the whole analytical workflow, from the sampling of these samples to the sample preparation that requires many different techniques, processes, and steps, to then the final analysis and quantification and the detection, determination of these micro nanoplastics in the sample. This whole workflow is really underdeveloped and not standardized. And another key thing that affects in this case, not to the analytics and the exposure, but to the hazard assessment, to the understanding of the effects, is that we lack real life materials. We don't have standards, we don't have representative materials that are really similar to what we have in the environment. And this means, if this is a picture of what is found in the environment, different types and sizes of micro nanoplastics, and this is what we are using mostly in the lab. We are using only one polymer, polystyrene, that's very monodisperse and with the same shape. So it's very different what we are testing, looking for the effects, and what is really found in the environment. So we have a problem here to really understand the exposure on one side and the effects in the other. So what we are trying to do in Plasticheal is to develop or at least advance in the development of the analytical methodologies, so we can really measure microplastics and nanoplastics in external matrices and in internal, in the body, inside the body. And we have also worked a lot to try to develop test materials that are beyond polystyrene in a size and shape that is representative of what we can find in the environment, and also that has a significant amount of particles below 1 micron. Once this is solved, then we can use all these developments to really measure the exposure and effects in a better position than we were before. So here you have an example of one of the materials that we have, for instance, generated in the frame of Plasticheal. This is PET from milk bottles. We have applied a protocol that is already published, and then we have characterized the material and demonstrate that this material is in a range of sizes able to reach cells. So we think that this could be a nice representative models. This is another example. This is PLA from TVAX. Again, we have published all the protocol that we have managed to develop to get micro nanoplastics below 1 micron that again are able, as you see here, to reach the cells, to reach the gastrointestinal barrier. Regarding some of the results on the exposure side, using FTIR imaging and confocal Raman microscopy and the workflow that we have defined and developed inside the project, we have been able to measure microplastics and nanoplastics in some external, the air, for instance, and foods, and also in human samples from workers in the textile and in the recycle industry. We selected these scenarios as we assume that they are more exposed than the general population. We still don't have complete information published, but I can advance that we are finding significant levels of micro nanoplastics in the air, in the water and the foods that we are analyzing, and also in the exhale raised condensate. This means nearby the lungs and the blood from the workers of these two types of industry. And regarding the effects, we have in several studies demonstrated that the nanoplastics that we are using are able to induce genotoxicity, so they can interact with DNA and cause alteration in this DNA. We have done this in vitro and also in vivo with the alternative model of Drosophila melanogaster. And as you see here in this example, we have significant, this is one graph from one study, this from another one, where you see that in a more or less concentration-dependent manner, we are seeing genotoxicity measured by the COMET assay. Here you see also some pictures. These are 10 images from the Drosophila larvae, where you can see the tiny plastics approaching to the what is the gastrointestinal barrier or the fly. They are really also close to the bacteria of the intestine. They can also then enter into the barrier through the microvilli, reach the microvilli area, the cell, and translocate to what is the equivalent of the blood from humans. So we have described the journey, let's say, of the plastics in this in vivo animal. And then in connection with this of the plastic being nearby the bacteria, we have also conducted some studies, and we have demonstrated that they are able to alter the microbiome composition. This is we are working with the larvae and also with the adult, and here you have some examples of results. This is the control. This is the positive control, and these are the plastics. And as you can see, there's a reduction of the biodiversity of these bacteria in the model, in the in vivo model, as compared to the control. And again, also the proportion of each bacteria that you see here in different colors. I'm not going to enter into the details, but you see here different colors. This is the control. Different colors means different proportions of the bacteria. And all the other cases, the positive control and the plastics, the color are very different, meaning the proportion of the bacteria are affected, are different. Moving from the gastrointestinal tract and the bacteria to the blood again, we have also demonstrated some effects in blood cells. We are using a system of exposing directly blood from donors. We have used polystyrene and our true to life, our real life materials in many different materials. And as you can see here, which is interesting, the different cells in the blood internalize these plastics differently, and the internalization levels of one plastics can be really different from another plastic. So what we have here is a really heterogeneous scenario depending on the plastic that we are using and the cell that we are analyzing. But regardless of this, if we analyze the things globally, we have demonstrated that these particles can produce reactive oxygenic species. They also alter the cytokines that are secreted for these by these cells, and they can also produce hemolysis in red blood cells. So globally, we have observed some potential immune dysfunction. And another important aspect of the results are the assessment of the long-term effects. As I have described at the beginning, the human exposure is it's a long-term exposure. So it is important that we also in the lab try to reproduce this exposure as much as possible. So what we do is that we have developed a system that keeps cells exposed to the nanoplastic through weeks. And then we freeze cells at the different points of the treatment. So we have a biobank of this long-term treatment with nanoplastics, and then we can analyze whatever endpoint during the process. And one of the endpoints that we have analyzed is the carcinogenic potential. We measure that with a battery of assays that measures features that are really connected to cancer cells. And then what we have found, as you can see in these pictures, and this is just a summary, is that after many weeks of exposure to nanoplastics, cells are able significantly to grow. As you see here, there's a cell growing detached from the surface. Only cancer cells can do that. They can also significantly migrate more. Again, this is a feature of cancer cells, and they can also move towards what is equivalent to the matrix, to the extracellular matrix. Again, also a feature that's only found in cancer cells. So we need to make sure that we assess long-term effects and the carcinogenic potential of these particles. Besides of the things that I've showed to you and the effects that I've shown, there's still a large number of unknowns. The dose where this analysis have been done, it's important because we need to make sure that all these effects are found also at the doses that humans are exposed to. So there are still many questions that we need to solve. And for that, of course, the only ways of proceeding is doing what we are doing here, sitting all together from our different fields to discuss and see if we can co-create measures to co-create the way of how to solve all these questions. And for that, with that, I would like to finish my talk. Of course, all this wouldn't have been possible with the work of my lab, the work of the Plasticheal team, the fantastic Plasticheal team, and also the cluster where we are working together for the same objectives with all the 4 projects, the ones that you see here. So many thanks for your attention, and if you have any questions, I'll be happy to answer now or at the end of the talk, of all the talks in the round table.
Thank you very much, Alba. I think it was really nice and insightful presentation. So again, I ask people in the audience if they have one very urgent question. And I don't see any. So I suggest that we move to the third speaker of today, and then we will have the general discussion as promised. So the third speaker is Dr. Gabriela Kalčikova, who is who obtained her degree at the Faculty of Chemistry in Brno, Czech Republic, and then the doctorate in Slovenia at the University of Ljubljana, where she is currently associated professor and carrying out her research. Her research team focuses on microplastics and the preparation and characterization of environmentally relevant microplastics in particular, with taking into account different aspects from aging to ecotoxicity, and let's say in parallel focusing on development of solutions that prevent microplastics from entering the environment. She's been involved in very many projects and international collaborations, including the preparation of reports with the European Commission and UNEP, focusing on the various aspects of plastics and microplastics impact. So again, you see already her title. So Gabriela, please, the floor is yours.
Thank you very much. Thank you for the invitation and for the nice introduction. So I think it's a good now connection with the human health and the environment. So I will now go to the environmental part. This is something that our team is researching for last, I think, 11 or 12 years. But when we are thinking about the microplastics in the environment, we have to first actually think how they are getting there, because this is also part of our work, how to prevent their entering the environment. So when we are thinking about the sources of microplastics, there is really a lot of them. Usually we are considering some plastic bottles or plastic bags that will get into the environment and they will start fragmenting, getting to soil or water. And this is one of the important sources, but there is even more. And we are contributing by our everyday activities, like doing laundry, when we are generating a lot of plastic fibers, artificial fibers that are released from textile. Also by driving a car, when the rubber tires are, of course, by abrasion disrupted and small particles are generated and getting like to the environment, like during every drive that we take. And of course, there are also a lot of synthetic paints, coatings on roads, on different surfaces, on boats, or as let's say protecting some different materials. And of course, because of the natural processes, they will also start fragmenting and getting into the environment. So I would say that there are basically infinite sources of microplastics, because every plastic item that we produced and used can actually become a microplastic particle. And when the microplastics are in the environment, it is also important to consider the transport. Initially, there were always ocean as a final sink of plastics and microplastics, but then a few years ago, it started to be clear that there is also a lot of microplastic pollution in different compartments, and going back from ocean to seas, to rivers, lakes, streams. The transport of microplastics is around the globe. So usually the first transport media is soil. So when they are found in soil, in plastic or microplastics, then they can get through soil into the, for example, groundwater, or with the surface transport, then to water, to rivers, and then further. In last years, there was also a lot done on air, which is really very important, because it seems that air is the main transport path, or the transport by air is the main pathway to get to dislocated locations, because we can consider transport by air very important for long transport and finding microplastics in location where they should not be. And there have been a lot of reports on these topics, where they found microplastics in desert, in Antarctica, and different mountains where there is no human activities. There are not any plastic production or use, and this is actually probably the main reason that there is the long distance transport. So let's say that now there is a lot of focus on air as very important, and wind as a very important transport media to different environmental compartments. But when we are thinking about microplastics that they are in the environment, we should not forget that microplastics or plastics, when they get into the environment, they are not the same. There is a lot of different processes that are affecting them. And we call this changes aging. This is also something that we investigate a lot in our projects. And we can have two types of aging in the environment. So the first one, it's biotic aging. So it is something that we maybe consider more the effect of UV radiation or the mechanical action. So when we have a bottle, plastic bottle, which is getting into a river or then sea, it is floating on the surface, on the water surface. It is exposed to the sunlight, and then it starts fragmenting, and it is producing microplastics that are again affected by UV or the mechanical action, for example, by wave, when the plastic bottle is disrupted, because it is also hitting the sediment and the sand, and then by the mechanical mechanical actions, it is then creating some microplastics. So this is the one aspect. And of course, it's important aspect because when it starts, the microplastics are exposed to UV or the mechanical actions, they have some cracks, fragments, smaller fragments are generated on their surface. So of course, their properties are changing. The second type of aging in the environment, which is also very common, is biotic aging, or you can find it under the term biofouling. And this is something that has not been so often considered, but it is very important because any material that is entering the environment is, and plastic especially because of their good adsorption properties, it is then exposed to biotic effects or biotic actions. So first, when the microplastic particle is entering the aquatic environment, then some organic and inorganic components are deposited on their surface, and this is a very good then starting point for microorganisms. And planktonic microorganisms, they can settle, they can attach, and they can start reproduction and creating a biofilm, because they will start producing extracellular polymeric substances, and they will actually cover the whole particle. And this is very important because this kind of particles, they are very different from those that are entering the environment. They have then different density. We know that most of the plastics that are used, they are lightweight, so many often they have lower density than water, so they can float. But when the biofilm is developed, the density is increasing, and then they can get into the water body. This is actually one of the ways why they are, for example, polyethylene, which is low density, and it should float on the surface. Why we can find polyethylene particles in sediment? Because of the biofilm and because of increase of density. But on the other side, when we have plastics that have high density, over 1, so then the biofilm is actually lowering a bit the density. So the biofilm is very important if you are thinking how the particle will be distributed in the water body. It is also important to understand that such particles, they are very often accumulated together because of the sticky biofilm. So also the size is increasing. There can be some aggregates together. And of course, it is now not only the particle, but also the biofilm and extracellular polymeric substances. So it means that also the adsorption properties are different. So, for example, more metals can be adsorbed on such a particle which is loaded by biofilm. And one very important thing is that we have also microorganisms attached. Here, I hope the video it is visible. There is a polyethylene particle that we aged for 12 weeks in our laboratory, and we found a rotifer attached to the particle. So it means that this organism can now actually travel with the very mobile particle. Otherwise, it would be very likely attached to some stone or plant. So with plastics, we also have to consider the possible co-transport of microorganisms and also some animals. When microplastics enter the environment, they are not only affected by abiotic and biotic processes, so it is not only about the aging of plastics, but they are also interacting with different contaminants that are in the environment. And research showed that there is a lot of reports about interacting of microplastics with, for example, metals, with nanoparticles, with organic pollutants. Many of them are priority organic pollutants. Many of them are very bioaccumulative, very toxic. And how microplastics and other pollutants will interact, it depends on the property of the plastic particle. It depends on the contaminant, and it depends basically on the surrounding environment. So it is important to know about the pH, about the ionic strength, and so on, because then we can consider if this adsorption of this contaminant on microplastic particle will proceed. We were testing this very often. We found, for example, lead very adsorptive on basically any plastic materials. We were testing polyethylene, polystyrene, polyvinyl chloride. We found also silver ions adsorbing, which is very important because, for example, silver is used as an antimicrobial agent, and it is also released into wastewater where we have also a lot of microplastics, so they can actually interact together. And one of our work, we were testing the adsorption of metal nanoparticles, and this was also related to possible co-occurrence, as again, the metal nanoparticles, they're used for a lot of cosmetic products and so on. So they can appear in the wastewater together with microplastics. And we could see that, for example, titanium dioxide is adsorbed very well. Nearly 70% of titanium dioxide adsorbed on polyethylene particles, while zinc oxide was not adsorbing very well. But the main reason was that from a zinc oxide, the zinc is actually released, and then we had more zinc ions in the medium than nanoparticles, and therefore the adsorption was probably much lower. So, but we also have to think about the interaction of microplastics and biota in the environment. And here we know that it actually can interact with any organisms in any ecosystems. It doesn't matter which feeding strategy the organisms have, if they are filter feeding, if they are predators. It is actually not very important. It is not important if they are aquatic or terrestrial organisms. Nearly for all organisms, they found that they can be ingested, that they can ingest microplastics, or they can interact via bioadhesion. And in this interaction, we can have a mechanical effect, chemical effect, or combination of both. So initially, I would say that in the initial microplastic research, the interaction of organisms and also with microplastics, it was more or less about the ingestion and about the mechanical effect. So it means microplastics, they were ingested by some organisms, and then there could be observed that there is some blockage in the gut, or there could be disruptions, there can be some mechanical problems. We work a lot with aquatic plants. So what we could see in our research that actually microplastics, they can attach to plants, aquatic plants, and they can also disrupt the roots or the membranes because of the abrasive effects, and they can also, so they don't have to be uptaken by plants, but also from the adsorption on their surface, there can be also some negative effects. But lately, I think the research moved a bit more towards the chemical effects of microplastics, because we know that plastics, they contain a lot of different chemicals. They are organic, inorganic. Many of them are very problematic, very hazardous. And of course, if they can be released from plastics, as we learned in the last couple of years, they can be released also from microplastics. Here is also one of results of one of our studies. We were working a lot on inorganic additives, and with help of laser ablation, we actually saw that we can differentiate microplastics even based on their metal content, because a lot of metals are used during production of plastics. For example, for polypropylene, aluminum is used as flame retardant, or you can use zinc oxide as biocide in PVC, or in polyethylene terephthalate. We know that we have a lot of antimony because it is used as a catalyst. So then you could see that it is not a very small amount, since it is covering the whole particle, or covering it is in the structure of the whole particle. And in our research, we also saw that it can leach very easily from the particles, especially when they are aged in the environment. So we were also testing a lot of different microplastics to see the leaching. So we were also testing first the effect of microplastics and the effect of leachate. So we could see that, for example, microplastics from bakelite that we were testing, there was quite significant leaching, and we also analyzed that there are some phenol-like compounds that are responsible for the toxicity. The leaching is, of course, it is not a rule when it will leach or when it will not leach. It depends on the on many factors, on how the organic and inorganic components got into the polymer matrix, if they are bound there or if they are only, for example, on the surface. And especially, as I mentioned before, the aging in the environment, it is really helping to release the additives. But of course, as I mentioned before, the contaminants, they can be adsorbed. So they can be first adsorbed and then they can leach and chemically affect the organisms. It is, for example, from this study, when we adsorbed first silver, and then when we exposed the plant, then we could see that those microplastics that had adsorbed silver, they were much more toxic, because very likely the silver was released and affected the plant. And we should not forget about the trophic transfer. So even if microplastics are only attached to the plant material, and even if they are not affecting the plant, then there can be some trophic transfer when they are eaten by herbivores. And this was also suggested that the organisms, very often, they are not able to discriminate between contaminated and uncontaminated food. So they are just feeding on their food source and ingesting microplastics. So the microplastics are passing from the plants to the next level. So as a conclusion, I would say that this topic is very, very complex. There is a lot of variability. And there is also a lot of interaction with the surrounding environment, changing of microplastics properties and ecotoxicological effects, changing their fate and transport in the environment. And I would say that it's also very important what we test, because as mentioned before, we are relying on spherical plastic particles, very often polystyrene or also polyethylene, but the plastics and the microplastics we have in the environment, they look very different. So as you can see the difference between the red polyethylene particle and environmental relevant polyethylene particle, which is a polyethylene fragment with loaded microorganisms. So it was after 12 weeks of aging in the environment, it looks like this, not like the red spherical particle. So I would say my advice is, don't forget to characterize your particles, because it is not wrong to work with different particles. It is not wrong to use spherical particles, but you need to know what are their properties and to understand how this will then affect the organisms or their fate in the environment. So thank you very much for your attention. This is the work of our group. If you are interested in our projects and in our work, you can check our very active Facebook, Instagram, and web page. And if you have any question, I will be happy to answer.
Thank you, Gabriela. It was another excellent presentation. So today, I think we really had three different perspectives that come together and provide testimony to the complexity of the field seen from, I would say, any specific angles, from regulatory to real environmental studies. So we are slightly late as usual, but I think it was worth it. And of course, there have been many requests to get access to the presentations of after this webinar is over. And I guess this week we will deal offline between the organizers, including myself and the speakers, if we can do that now. And then we have content information for all the participants, so we will let you know. Coming to the science, let's say I'll try and start with some of the more general questions in the chat. So first question is that I see is what challenges do the difference in scale between micro and nanoplastics, particularly nanoscale, pose for analytical methods, which I guess means how do you use different analytical methods as a function of particle size. I guess this is general, so any of the three speakers who wants to pick it up?
So maybe I can start. So yeah, it is a very big difference between nanoplastics and microplastics. We do a lot on preparation and characterization, and to prepare environmentally relevant nanoplastics is really difficult. And it is also difficult then to monitor them, and it is even difficult to monitor microplastics. So definitely it is understandable why research on nanoplastics is using the spherical particles that are characterized and that are produced. In microplastic research, I think we can now already afford production of our well-known, very well fragmented particles. So I would say that this is the main difference, and the analytical approaches are a problem because, as Alba mentioned, to follow where there are the microplastic, where there are the nanoplastics, it is not very easy. It is not very easy, and even in an environment when you imagine sampling even clean water and to find their microplastics, is this difficult to get to, for example, 10 or 5 micrometers, and for nanoplastics it's like really I would say to find nanoplastics in environmental metrics, it's by chance.
Alba, do you want to comment?
I'm not really expert in the analytical methodologies, so I can really go deep into the answer more. But yeah, I agree. I mean, the protocols are different. The techniques that you may need to use for one or the other are very different. So you have to work on the workflow for one or the other separately. As I said, inside the project, we have managed to really measure nanoplastics in water, in foods, and also in blood and in exhale rate condensate. But it's really complicated, time-consuming. You need a dedicated expert. I mean, it's not something that at the moment we can do, let's say, for monitoring. That's completely impossible. But at least we have managed to do that for research objectives. And I guess that we'll see in the coming years how this evolves, because it's really a key thing in the field.
Okay. Then I have a more specific question for Silvia regarding the process and timeline for updating an ISO standard. Can you update it?
I can confirm that in any moment an ISO standard can be revised. So again, the normal life cycle is 5 years, but if in the committee at the national and international level there is the request to revise, it can be open a new work item, can be identified a project leader, and the ISO standard process can be started. So in any moment can be revised.
Okay, that's interesting to know. And I also see a question on the Q&A thread regarding the type of techniques for the characterization of NPs in cells. So can you comment on the techniques in vivo, let's say, from a more general perspective, maybe? I don't know, Alba, you want to go first?
Yeah, sorry. Can you please repeat? I was distracted reading some questions in the chat. Sorry, Antonio.
Yeah, no problem. The question was, I was wondering what type of technique were used to characterize the NPs in cells.
Yeah, I already answered to the question. It's confocal microscopy. And for that, of course, you need to label your particles. And this is another challenge. Because if you want to work with true-to-life materials, real-life materials, they do not come fluorescently labeled. So you have to do something additional to be able to track them into the cells, which, by the way, we consider fundamental. It's really important that everybody demonstrates that the particles that they are using are really able to reach the cell. So there's a connection with the effect that they are observing and the exposure of the system. So we have also investigated and developed some method to fluorescently label the particles, the real life particles, and then with that we can then assess internalization, exposure. I have provided in the chat the reference. Just if anybody is interested, you can go through the article.
Okay, lovely. Thank you. So the other question I see is relatively long. I'll read it through. So it says, given the complexity of different environments, how reflective could future materials we use to study MNPs in the lab be of these environments? For example, could it be possible to generate MNPs with biofilms in the lab in the future, and we use specific weathering techniques too? So Gabriela, I guess this is.
Yeah, so we actually generate microplastics with biofilm in laboratory, because if you would like to have a biofilm on microplastics from the stream, it is very likely that you will not find your plastics there anymore after some time, because it is just because of the flow and extreme conditions. It can happen that they will just disappear, and of course you have to put them to something that they will stay. So some a bit larger microorganisms, sometimes they cannot get inside, because we are working with microplastics, for example, from, I don't know, 10-15 micrometers up to 1 millimeter. So there is very small particles that would disappear. So what we do, we are using a natural water, and then we put the natural water into Erlenmeyer flask. We add our microplastics that we prepare from consumer objects. So we can we generate microplastics that would be generated in the environment, and then we age them. We just leave it, shaking it, and we just replace the water every week, just to be sure that we have enough nutrients and enough microorganisms. And the pictures that you saw of the very nicely covered particle, it is from a laboratory, and they are very similar to those that you can find in the environment. So it is possible. We have a protocol, so if someone is interested, I can share it. I can share it with you, and you can start producing your environmentally relevant particles in your lab. It's very easy.
Okay. If I add something to this, well, actually, Gabriela already commented, no, the thing at the size. No? At present, it's possible, and there are published protocols of obtaining degraded plastics or micro nanoplastics from the degradation of plastic items. A bottle of milk, a tea bag, or the other items that Gabriela, for instance, is using. But going to the environment and getting micro nanoplastics, that is a completely different thing. And if you think on health perspective, I already mentioned that the only ones that matters, or well, not the only ones, but the ones that matter the most to health are the ones below 1 micron, the nanoplastics, and those are not possible to collect in sufficient quantities to really apply this to a laboratory experiment, and sufficient quantities for also for other laboratories to test the same materials and see if they observe the same effects or not. So at the moment, the only possibility is to really produce microplastics and nanoplastics, which are important for health, from plastics items. There's a chance to do this, but
it's not possible to extract actual microplastics from the environment and then to test them, because even if you want to extract them, you have to apply a procedure which is affecting them, and it would probably remove the biofilm and so on. So we are trying always to get to the very environmental relevant microplastics, and I can see that, Alba, this is the same story, that you are trying to avoid the spherical plastics with anything. So plastic particles that actually can exist in the environment, or can be actually hazardous for human, or at least the human can be exposed. But really getting them from the environment, it's currently impossible, and I think this will not change really a lot in future. I think the way how to deal with this is to sample and to see what we can find in the environment and try to mirror it in the lab.
Mm-hmm. And if I may say, also, no, if you think from the microplastics perspective, we're talking about the biofilm, no, and the interaction with bacteria, which is certainly interesting. If from the microplastic perspective, if you have a microplastics, different bacteria can attach there and form a biofilm. But if you have a nanoplastics,
they will be more in bacteria.
The bacteria is bigger than the nanoplastic.
Yeah.
So it's the other way around. It's the nanoplastic that is attaching to all the surface of the bacteria, having an impact on its potential behavior. And talking about health, this also, of course, goes linked to the microbiome disruption, and also what could happen, for instance, to pathogenic bacteria, so the bacteria that enters into our lungs and produces whatever disease. No, if the bacteria comes with the microplastic or joins the microplastic once in the lung, what would happen with the behavior of this bacteria? Would it be more that hazardous or less? Or the topic is interesting, but again, there's a completely different perspective if we think on microplastics than if you think in nanoplastics. It's something that we need to really consider.
Yeah, thank you. That's a very interesting perspective indeed. And I would like to prioritize the last question, since it looks more general, regarding the standardization and the use of real plastics, which I guess is environmental samples. Can you standardize that? So the question is, are real plastics difficult to standardize? And also from the animals side, what are the perspectives for standardization of animals or small animals, I guess, for mimicking the exposure?
May I reply?
Sure.
Not exactly to that, but is in the experience that I've seen. Of course, to standardize an in vivo experiment is not easy. What we have to focus is to standardize the method that we use. So whatever is the situation, that could be different environmental situation. The example that Alba brings before, so how the biofilm is built up on the plastic, or is a microplastic, nanoplastic inside or outside the bacteria, and so on. So now we have to focus on the method that we are applying to identify the microplastics. This is the very first level. And after the effect of that these microplastic have in vivo, this is another step. And the standardization in that is really different approach. What we should focus at least at the beginning is to standardize the method to identify the microplastic. And listening your presentation, I think that for sure could be the image analysis extremely useful in terms of we have to pay attention because frequently the image analysis considered the algorithm that the system already provide you. So the manufacturer approach to you and say, okay, this microscope can calculate and count how many particles and so on, so on. But you have always to have in mind.
Silvia, sorry, we cannot hear you.
Oops, can you hear me now?
Yes, yes.
Okay, sorry. So you have always to consider that the pure image analysis. So you can record the image and you can share with your colleague and you can study that. In that way, we get a great experience. We use the image to train the technicians. And after this training, we obtain a much better equivalence in counting or in doing some microscopic method, for example. But so really, I would like to say at the moment to maintain the standardization level to the method of analysis. So to check the microplastic, and regarding the effect that this microplastic have, this is another level of discussion.
Yeah, and regarding this, it's really tricky to get standardized materials or reference materials, or you can call it in different ways. Because for that, you need to produce the material in sufficient quantities. You have to store or resuspend and keep the materials in a way that is not prone to contamination and having traces of many other things. And then you have to distribute this as a reference material to many labs. And this is currently not possible. The stability of the material is tricky. The maintaining the material without contamination is tricky, and the quantities that you can produce with the protocols that we have now is not adequate. We cannot really do that at the moment.
Why not to consider it an artificial material, not as a reference, but for example, sorry, it's just an example. I'm trying to compare to the other situation. You have an artificial material, and this artificial material is homogeneous, is stable, is distributed to the other lab, and it is added in a certain amount to a matrix, and you have to recover that.
Yeah, I know. For the analytics, it's a completely it's a different. I was going in the side of the effect side, no, because for instance, because maybe for the standardization of the analytical tools, you may be able to, because the concentration of particles that you need for standardizing this, and it's not really big. So you can maybe do an exercise of standardization, validation, interlab comparisons with the methodologies. That is possible. But I'm thinking on the other level, like having a material that's useful for references, for analytical methodologies standardization, and also for the effects characterization. At present, we have, I don't see how. I mean, I wish this is all what everybody in the field really would like. So everybody can compare results with the same material. But it's how can we manage to do that if we cannot really produce something that it's always the same in sufficient quantities and stable to be distributed? But it is, I mean, it's of course it would be a really nice, I think, to have.
In agro-food, we use a lot of to lyophilize the material. So the lyophilization process, if you have natural matrix, the lyophilization produce a very stable material. But again, so just an example.
No, we are. I mean, we are discussing about the two main important things in the field. It's the analytical tools and the reference materials is the two main things that are really the most challenging. And there are many others because also the models, the in vitro models, the in vivo models that we are using need also adaptation to the exposure to the plastic. There's many other things, but of course these two are really common for everybody working in the field. People that's working in the exposure, people that's working in the effects, human health or environmental health. We have these two common challenges.
So since time is running, I just wanted to ask each of the three speakers a very brief statement regarding what is in the scope of the project. So essentially the availability of, what's their opinion on the availability of services from the European infrastructures. Is there sufficient capacity? Is the, let's say, portfolio of services reasonable to support, I mean, other people's research? Or any of your thoughts would be interesting.
So maybe I can start. I think it's really important to create some kind of network with and to describe the capacity of the organization, what they can do, because I see there is a couple of institutions that are able to produce microplastics and well characterize them, but I think it's maybe not enough, as there is always a lot of question and a lot of interest for to produce environmentally relevant microplastics for other groups. But I think yeah, we would need a bit more, and maybe really to somehow better organize this effort, because all the researchers on microplastics, they need microplastics, but not a lot of them are producing them, and not a lot of them are properly characterizing them with everything that we need to know about the particles to understand their effect and fate. So definitely I'm really happy about the FHERITALE project and about your activities, and I really hope that it will be very successful.
Thank you.
Yeah, well, if I can add something, I mean, it's important also to keep in mind all the effort that has been done before with the nanomaterials, because there's a lot of projects that have been ongoing for the last years about this, and they have advanced a lot in the characterization of this material. They have done a lot of validation, standardization, etc. So it's important, my wish would be, that we really move on things that we really don't know and cannot be really applied or adapted from the nanomaterials to the plastics. If there's been efforts before us, before the project that we are running now, that have already solved some of the questions, let's make use of the data that have been already produced by the community and by really fantastic projects that have been developing in the nanomaterial world.
Thank you, Alba. Silvia, do you have any comments?
No further comments. I understand as it was my impression, it's a very interdisciplinary topic, and it requires a lot of expertise and well connected. For sure, there is already a lot of material to consider it, and but I've seen that you have already done an inventarium or a list of what you have. So I really hope that I'm sure that, I mean, the project is structured in a way that should facilitate a lot the comparability or the equivalence, let's say.
Mm-hmm. Okay, I think we spent some more time than anticipated, but I really think it was totally worth it. So I would like to thank our speakers again for their very interesting presentations, and we will get back to the three of you for this item of the distribution of presentations, if possible. And again, also thank to all the participants for joining us and staying even longer than anticipated.
Okay, thanks to you.
Thank you.
Thank you for organization and invitation.
Thank you very much.
Thank you. Bye.
Bye.